and could not really spend much time in discussion afterwards, after the class, because although we are doing a lot of discussion in the class, so that's during the lecture, so that's okay. But more specifically, we did not really got a chance to see how one can design studies or what we can do with them. So what I have done is following, that for each class, like yesterday we had we did some sort of studied normal and cancer cell divisions, we did some epidemiology. Today we did extensive cell cycle analysis and how the regulation of cell cycle is occurring, and we also did some growth factor regulation. So we spent like couple of hours on cell cycle regulation. So instead of my continuing to expand on the same thing, because I think I really pretty much covered the apoptosis, growth factors, and the cell cycle regulation. You can go into much more detail, but I think that for our practical purposes here, I think what I gave is sufficient. So now then I would be, if I have some slides, but then what I would be doing is giving more like a research paper, and that's not what you want to see. So this is like general concepts. So what I will do in the afternoon session today and tomorrow is show you some small video clips, and they are not our work, they are not from my lab, but what it is from the YouTube, and I've selected from wherever we are. Tomorrow I'll show you one small mammary gland organ culture video as to how the glands are dissected out and things like that. It's very tricky. That we'll do tomorrow. So today I'll have maybe 3 or 4 videos for about they're not very long, couple of minutes, 2-3 minutes long, some of them are 1 minute long, but then that will open up our discussion session, and we'll see how the discussion goes. When you guys have no questions, then we'll end the session, or I can continue going into the next class, but I think it will be kind of a too much overload, it's a material overload, and we don't want to do that. So you can digest it a little bit what we did today, because the regulations we discussed were quite intense today. So right, okay, so what I'm going to do is, first I'm going to show you the one with normal and cancer cell, how do they grow in culture. So this is a time-lapse video done by National Cancer Institute, and they had put it on their website of the National Cancer Institute in the USA. A very small video, but it shows how the cells actually behave in live situation in the culture. Okay, and then we'll be talking about the epidemiology and the smoking. There is a lung cancer video, and that one is really explaining just in nutshell what we talked about yesterday in detail as to what they are looking for, what are the validities, what are the statistical, what's the importance of statistics in epidemiology. And then we'll do cell cycle analysis. I have one on that. Yes, cell cycle regulation. We'll do that, and then we'll do one of the pathways, signaling pathway. So we'll do 4 videos. Okay, so let's start driving. Okay, first is the cell culture, normal and normal and cancer cells in culture. Okay, how do I? Oh, yeah. farther, the cancer cells are very small in that area. You can see the cells started piling up. You can see they're not, no. You can see, you see this area. Okay, it's almost over. Okay, so what we wanted to see was that, for example, the couple of things. The normal cells have the structures that it maintains many cells in its own growth. Even when you start out becoming a small cells, eventually it elongate, become fibroblast. That characteristics is for many cancer cells also. They become fibroblastic in nature. Fibroblasts are long cells with a central nuclei in them. One of the phenomenon is that normal cells, as we talked about, has a very intact contact inhibition. It does not pile up, whereas cancer cells, you saw, they just kind of jumbled up. Now in this video there was no nuclear the shrinkage and things that were shown. I was trying to find that video, but I really did not see that much. So this video is done by the National Cancer Institute. It's too bad that they really did not have much of an explanation with it as to what kind of cell lines they used and what kind of details that they were looking at. But I think that we really don't need to do that much on this. I'm going to show you the cell cycle regulation video, because that's what we talked about today. Well, epidemiology is 1 minute video. Let's watch that also. So you can just
The human body can be exposed to many harmful substances. For example, we know that smoking causes cancer. But how do we prove the correlation between the substances taken into the body through smoking and the damage that results?
Epidemiology, which literally translates as the study of the people, plays a critical role in establishing the cause of a disease.
Today we're going to see a really simple activity that helps students understand the concept of correlations. In particular, examiners will expect students to understand that correlations cannot show cause. The other issue we'll be looking at is the impact of confounding variables and what other factors we have to consider when carrying out correlational research. In psychology, two of the most important concepts are the concepts of reliability and validity. We need to be sure that our research is both reliable and valid. By reliability, we mean the consistency of our findings. When we're talking about validity, we want to know if we're actually measuring what we set out to measure. Students often find these concepts difficult, so what we're going to do today is show you in a very simple demonstration how these concepts can be made more understandable and accessible to students.
Okay, that's all very simple. So what they were saying is that majority of the epidemiological studies, as we said, based on correlation, but then we went into much more detail of deciding to use the ratios, the odds ratio, the relative relevance ratios, the RRs, and so on. So what it is that the statistical, two things you have to remember: statistical studies are huge, and if you don't have enough samples, you need to have many institutes get together within the same area or distant areas, and they all have epidemiological results. You combine them and then you do a meta-analysis type of study, and those are usually those are respected more by the journals also, because they are independent results, and they're coming from too many directions, too many people are talking about it. So the meta-analysis is also a more stricter statistical analysis. So there are also chances of not getting results that will provide you for significance, and the reason is that different institutes may have used different exclusion criteria. In epidemiology, one of the things that you really need to know is the exclusion criteria. For example, if you're looking at the people with say breast cancer, so you want to take into account people whether they're taking birth control pills, they are on hormone replacement therapy, they were on all kinds of hormone related issues, they all are cancelled out. The age is important, so whether you're taking certain age, whether you're taking the earlier age, you're taking the older patients, so you have to cut off the limit, saying, well, we'll select only 55 plus. So what happens is, for example, I'll give an example for a tamoxifen. In a tamoxifen study, which was done by huge number of people, normal people in the United States, they're all nurses. It's called nurses study. So they have followed the habits of people for many years. They got the tamoxifen treatments, not the treatment, just the effect of tamoxifen is more as a preventive approach to have the prevention, and found whether there is any activity in terms of cancer prevention or not. The results came out that okay, tamoxifen could serve as a preventive agent, but not as efficient as they had originally found. So things like that are important. The statistics is the number one important thing in terms of studying epidemiology. So people who do the epidemiology, they usually have a health science masters or public health masters, where their major emphasis is statistics. And like for example, majority of us, when we do the analysis, we just kind of run your standard deviation and correlation coefficient and just show the standard error bars or standard deviation bars. But in epidemiology, that doesn't work. It's much more in-depth, detailed analysis. Okay. So now, and then we'll open up general for discussion for all the subjects combined. I will show you the cell cycle regulation video, which we talked about today.
Cell growth and division are divided into phases. Cells grow during the G1 phase, and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclin starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again.
I thought he was going all the way to 3 minutes. I haven't seen this video either. Okay, I think he's done talking. Okay. Why is not? Oh, I have to turn it off here. I see. Okay. So that's cell cycle regulation, and then I have one signaling pathway. Actually, I have two signaling pathways. So let's see what they have. The two of them. We talked about the PI3K, and we also talked about the MAP kinases. So we'll see what we have here. Let's see this one.
In response to a cellular signal, such as ligands binding to a cell membrane receptor, phosphatidylinositol 3 kinases, or PI3 kinases, catalyze the transfer of the gamma phosphate group from ATP to the hydroxyl group at the 3' position of lipid substrates. Signaling through PI3 kinase activity modulates many cellular processes, including cell growth, gluconeogenesis and glycolysis, and motility and differentiation. There are 3 classes of PI3 kinases. Class 1 PI3 kinases catalyze conversion of phosphatidylinositol 4,5-bisphosphate, or PIP2, to phosphatidylinositol 3,4,5-bisphosphate, or PIP3. Production of PIP3 by PI3 kinase is antagonized by the activity of tumor suppressor tensin homolog deleted on chromosome 10, or P10. PIP3 binds to specific domains, such as the plextrin homology domains of target proteins, and recruits those proteins to the membrane. AKT and phosphatidylinositol-dependent kinase 1, or PDK1, are kinases that with plextrin homology domains bind PIP3. At the membrane, PDK1 phosphorylase AKT at one of the two required sites for AKT activation, threonine 308. The monophosphorylated AKT is released into the cytoplasm, where it interacts with the mammalian target of rapamycin complex 2, mTORC2. The mTORC kinase component of the complex phosphorylates AKT on serine 473, and now the dually phosphorylated AKT is fully activated. Fully activated AKT is free to interact with a host of substrates. It phosphorylates and negatively regulates tuberous sclerosis complex 2, or TSC2, leading to the activation of the small GTPase, RAS homolog enriched in brain, or RHEB, and subsequent activation of mTORC1. Active mTORC1 can enhance cell growth and proliferation through its interaction with 4E binding protein and ribosomal S6 kinase. AKT can also translocate to the nucleus, where it phosphorylates FOXO transcription factors, leading to the expression of genes involved in oxidative phosphorylation and energy production, upregulation of chaperones and heat shock proteins, and the production of antioxidants. Activated AKT can also interact with pro-apoptotic proteins, reducing pro-apoptotic signaling in the cell. AKT also influences metabolism, nitric oxide production, and angiogenesis.
Okay, so what happens is that this kind of tells that what we wanted to do in the class was to give you really an overview of how the growth factors or how the transduction, signal transduction works. Now as you go into the detail, then you have, like in this one, it was shown on the PI3K pathways, which are really important for many carcinogenesis. You can actually select out the carcinogenic process, for example, PI3Ks and the colon carcinogenesis or breast carcinogenesis is kind of very much correlated. Now the use of AKT, or in signaling of all these processes involve many proteins, as that video was saying, but that's kind of beyond the scope of our presentations here. So we did not go into any selected molecule. You can do that, but then just showing you how the molecules behave, but that's not the intention. What we want to do is to give you a global view of how the signaling works and then how it helps design our experiments, because we are all kind of at some stage of research where you need to know how the new experiments are designed, be it drug discovery or signaling or understanding of regulation of some sort of a compounds or activities and so on. So that's the sole purpose of this. I'm going to show you a MAP kinase signaling pathway, because there are three of them important: beta catenin I told you, AKT I told you, and the one that is MAP kinase. So we can watch MAP kinase.
The MAP kinase signaling pathway. The MAP kinase signaling pathway in the normal cell. The designation MAP kinase, abbreviated MAPK, originates from one of the first members of this protein family, the mitogen-activated protein kinase. The MAP kinase pathway begins with the binding of a ligand, for example EGF, to the extracellular portion of a membrane-bound receptor of the family of receptor tyrosine kinases, abbreviated RTKs. Ligand binding leads to the dimerization of two subunits of the receptor tyrosine kinase. At the inner side of the receptor, tyrosine kinase domains catalyze phosphorylation of itself and also of the other subunit. Next, the growth factor receptor-bound protein 2, GRAP2 for short, can bind to the phosphorylated RTK. The protein SOS, named after its homolog son of sevenless in Drosophila, is able to bind to the membrane-bound protein RAS. Inactive RAS is bound to the nucleotide guanosine diphosphate, GDP. SOS catalyzes the exchange of the RAS-bound GDP against GTP, guanosine triphosphate. This exchange leads to the activation of the RAS protein. In its active GTP-bound state, RAS is able to bind to several effector proteins. The kinase BRAF is one of the most important effectors of RAS. Active BRAF phosphorylates and activates the kinases MEK1 and 2, which in turn phosphorylate and activate the kinases ERK1 and 2. Finally, the kinase cascade leads to the activation of transcription factors of the AP1 family. The abbreviation AP1 stands for activator protein 1. The best known AP1 proteins are the transcription factors Jun and Fos. After their activation, Jun and Fos move to the cell nucleus, where they form a heterodimer and bind to an AP1 motif of the DNA. This leads to the expression of many genes encoding, for example, growth factors, cyclins, and cytokines. As a direct consequence, cell proliferation is activated. In the normal cell, the active RAS-GTP complex is inactivated shortly after its activation. In this way, a permanently active MAP kinase signaling pathway and undesirable effects are avoided. RAS-GTP is inactivated by the GTPase activating protein, also known as GAP. GAP binds to RAS-GTP and increases the very weak GTPase activity of RAS by several orders of magnitude. GAP achieves this activation by providing a domain that assists the RAS protein in the hydrolysis of GTP. The bound GTP is hydrolyzed to GDP. GDP-bound RAS is no longer active and can no longer bind to BRAF. As a result, the MAPK signaling pathway is turned off. The MAPK signaling pathway in a tumor cell with RAS mutation of the RAS gene may have dramatic consequences for the cell. Mutated RAS protein is activated just like the normal protein. Mutated active RAS-GTP is also able to bind and to activate the kinase BRAF. A major difference between the mutated and the normal RAS-GTP protein is that the mutated protein loses its capability to be inactivated by the GTPase activating protein GAP. GAP can still bind to mutated RAS in its GTP-bound form. However, GAP is not able to provide the domain that is crucial for the activation of the RAS-GTPase. GTP is not hydrolyzed to GDP. Consequently, mutated RAS-GTP stays active. The kinase BRAF stays bound to RAS-GTP and remains active. BRAF phosphorylates and activates an increasing number of MEK1 and 2 proteins. In turn, MEK1 and 2 activate ERK1 and 2, which activate Jun and Fos. Thus, the kinase cascade is not turned off. A permanently activated MAP kinase signaling pathway results in continuously activated proliferation, which is an important feature of a tumor cell.
Okay, I think this is a good video. So this one really sort of goes in a little bit of a, what you call, the animation that we talked about. We did talk about GAP and GED, how the RAS becomes activated, and the in the presence of GAP it becomes inactivated. So those are the major ways that it happens, like the RAS activation, and then those GAP and GED are not really going to be constant for all kinds of growth factor. I mean the oncogenes, but for RAS it's been established, right? So yeah, I think that my videos. There's one more. If you want to go, I can show you this one. This one is pretty good too. Is what are the best practices for IHC detection, immunohistochemistry, and then we can open up for discussion. Let me see one more video. Okay. So because we could not show it yesterday, I had 2 and 2 every day. So
First of all, I would like to thank everyone for taking time from their busy schedules to join me today for this webinar on best practices for the immunohistochemical detection and interpretation of estrogen receptor protein, progesterone receptor protein, and HER2 protein overexpression in breast cancer. Last year and earlier this year, I presented a longer 3-hour version of this for training purposes for, I like to use my friend Dr. Clive Taylor's total test concept, where the immunohistochemical technique is broken up into 3 compartments: pre-analytic, analytic, and post-analytic. Hopefully today I will have enough time to discuss the items in blue: specimen type, acquisition, transport time. A very important issue today is fixation for breast biospecimens, and tissue sectioning, which does not get
because many of you are doing immunohistochemistry.
And then moving into the analytic compartment, we'll talk briefly about personalized antigen retrieval, our testing protocol here at Clinical Laboratory Partners, Hartford Hospital, and a little bit about reagent validation. And then finally, we have the post-analytic compartment, which is evaluation of controls, interpretation of results, and reporting these results.
Not very clear.
Now,
you can see the picture.
Here we have a diagnostic core biopsy, and you can see the invasive carcinoma, the invasive duct carcinoma. Actually, I should say invasive carcinoma, showing an absence of myoepithelial cells as demonstrated by a lack of smooth muscle myosin activity. And those same tumor cells.
No, I'll find some other video which is immunohistochemistry. I had seen this videos, but I selected them based on thinking that what they would show, because immunohistochemistry I thought is very important because many of you are using it, and there are some practices that are very crucial for immunohistochemistry to avoid backgrounds, to do the antigen retrieval, to how to determine the time of antibody exposure, how to determine the antibody dilution. Many times you want to dilute your antibodies way like 1,000 times, and if you do it more concentrated, you're going to get really lousy results rather than having it a very diluted antibodies. So some of those parameters are very important in terms of determining or doing your immunohistochemistry procedures. So I thought I'll just show you, but you can't hear too much in that. So let's talk about what you guys do and what you have problems with in terms of studying this kind of is anybody doing cell cycle analysis? Looking at the cell cycle now, or growth factors, or anything that we did today. There is no anybody's interested in doing it tomorrow. In the near future. So because that's the direction you guys are going to go. If you just, my feeling is that if you just look at the activity of extracts, which most of you or many of you do, if you don't go a step further, it's going to be a rude awakening that's not going to be enough, because we know that extracts, analyzing or evaluating extracts is very important, but that's only the beginning of the process. I'll show you one slide which will explain that very easily. If I can find it quickly on my, it won't be, I don't know that's in here. I'll show you. I was going to talk about that slide later on, but let me show you if I can find it. Because what happens is that what we do really is, where is my GIAN? It's on the, okay, right here. This is the one that we're going to be talking about in chemo prevention, but I'll just show you one slide. I want to show one slide. and so he will grow there some plants and then generate extract. Sometimes he'll bring the extract from there. Now this whole project was based on the bioactivity guided identification of new cancer preventive agents. So what we do is that they take this extract and run into 7 or 8 different assay systems. So they will do the anti-mutagenicity, do the antioxidant property, look at the hormonal suppression, look at the receptor profiling, they will look at the PKC activity, a bunch of them that are related to various cancer forms, and they will do all these activities on this one extract. Now they'll say, okay, this extract looks active, and that looks active in, say, a MCF7 cells, which is a breast cancer cell line, but it did not show any activity in antioxidant type of reasoning. So you already know that this plant, when we get a compound, it will have something that will be anti-estrogen, maybe, or anti-progestational. So it's called a bioactivity directed fractionation. Okay. So now, once the active is found, then it has to go through scrutiny. So we do the MMOC. I'll explain to you. MMOC was developed in my lab. Actually, it was developed by me, as that was my PhD thesis, actually. So I've done few thousand of those mice. So then what happens is this active extract gets evaluated in the mouse mammary gland organ culture, and that's not sufficient. So I'm going to go explain in detail later on, but what it is, just briefly, a few liner, is that mouse mammary gland organ culture, when we culture it it's an organ culture, not cells. So you take the entire organ from young animal, put it in the culture, and if you give proper hormones, it differentiates like the mammary gland in the animal. So if you give like the hormones that produces milk, it will produce milk. You will have casein expression, alpha-lactalbumin expression. If you put prolactin, it will induce like lot of RNAs and the one that you can have genes and whatever prolactin inducible genes. Things like that, you can do everything in there. If you remove all the hormones, it will regress back to the normal stage. So what that means, and I can do that in 10 days. So if you look at an animal, so the animal, when the animal goes through from virgin stage to pregnancy to lactation, the gland structure changes. Just that's the model we use. Just also in women, like for example, with the mammary glands growing, the proliferation is very high, right? After lactation, the gland ceases. In the animals, it becomes back to the normal stage, like a young animal stage. So if I remove all the hormones, it becomes back to that. Okay. While doing that, on the day 3, if I put a carcinogen in there, then it will grow just like in the presence of hormones, just like normal mammary gland would. But then when I remove all the hormones, instead of regressing to a ductal stage, it will show a lesion. It will show some areas that are not regressed. And those unregressed areas, if you put it back into the animals, after 2 or 3 generations, they'll form tumor, cancer. Okay? So that's the model. So we don't go all the way up to cancer stage, but we'll say, okay, carcinogen and carcinogen plus your compound. And if you do not develop those lesions, which you do in the presence of carcinogen, that means you have some activity of that extract. Right? So that's the process, that's the model. So everything has to go through my model, and we have done many, like several thousand MMOCs, and it's like my technician, you'll see him, you'll see his hands. there's a video I'll show tomorrow taking out the mammary gland. So I just tell him that, you know, both of us are going to go to hell because we have killed a lot of animals, but at least I'll have a company because you're going to be with me. So we'll do MMOC. So now we have a lead plant. Now that lead plant, then it comes, this is a duck kinghorn, my friend. He is going to fractionate all this extract. The active extract will be fractionated by using HPLCs and all that into various fractions, and they collect all the fractions. And those fractions get evaluated in the bioassay that was found active here. Only one assay. We don't use all multiple assays because you'll have like 50 fractions. So you cannot evaluate all of them in all the assays. So you see that the active assay, and now you have an active fraction, okay, from this one. And then you process and say, well, okay, this fraction has a possible chemopreventive agent because that is active. So we try that again in MMOC. So that's a second level screening, a higher level screening. One level screening is cells, but now you're going to the organ. And then I also do the ACF. ACF is the aberrant crypt foci in colon. So that's a predictor for a colon cancer. So and we have numerous papers in this also. So we do the mouse mammary gland organ culture and ACF on this chemopreventive agent. If it is active, it's an active chemopreventive agent. Anytime it is inactive in MMOC, like for example, if it's not active here, that plant is shot. We never look at it again. If active, the fraction is not active in one of these two assays, that's it. That's the end of it. If it's active, then we'll have again from here we identify new chemicals. Like isolate the chemical, the medicinal chemists will give it a name. I still haven't figured out how they name them, but they name the compound. Like there is a compound that I had is called brasinin, which we had identified a long time ago, and published a really good paper. At the same time, sulforaphane came along. It was the same kind of compound. So brasinin was indole, sulforaphane was a little different chemically. But then these medicinal chemists give it a name by there was one hydroxyl group was less in a compound, it was more active. They called it tefrosin. Now, how did they get from brasinin to tefrosin? They cannot explain to me. I said, how the name is, how they name the compounds, that I don't know. But they call it some name, and that gets evaluated in the same assay, and it goes through this process, and then you have an active chemical, active compound, which has a name. And then what we do is, then we have a Moriarty, Robert Moriarty, worked with very, he's retired. All these guys retired now. They all have, like me, Moriarty is retired, probably much older than me. And but he had a large group. He's a synthetic chemist, but he worked with 2 Nobel Prize winners. So he's a pretty sharp guy. So he will make analogs, and he made my vitamin D analog. So the one which I'm going to talk about, which is in clinical trials. So he synthesizes different analogs of this chemopreventive agent, and then that gets into the testing. For example, all the bioassays. But it also gets synthesized in large amounts. So we can do experimental carcinogenesis in vivo to see if it is active. If it is active in here, then we study mechanism of action and say how it is done. So now we can write a grant on that compound from government, say that well, we have identified a brand new drug and we want money to go take it to the clinic. Well, that's the way to go forward to have some sort of a group with the graduate students, postdoctoral fellows, and then we can expand onto that if you have funding coming, and then you can go into that direction and study more mechanism of action. Once the compound, once if it looks very active in the in the chemical carcinogenesis models, which I'll be talking tomorrow, then what happens is that we have to do one more step, which is a preclinical toxicity. Now, the company, the research institute I come from is known for its toxicity studies. That's all it's known for in the whole USA. They do millions and millions of dollars of business with industry. It's a CRO. It's a contract research project. So many companies will give them their compound. The only thing is, it's very expensive. One compound to go through the toxicological study costs about $500,000. So, it has to be GLP, which is a good laboratory practice. You have to sign and double sign. You cannot cross anything. You cannot cross any number. You have to have signatures and all that. So after that preclinical toxicity is done, then it can go to then it can go for FDA approval, and then phase 1 clinical trials. Once phase 1 clinical trial works out, then you have a cancer preventive agent. Okay. So what I want to go through this to really emphasize the fact that having worked on extract, you have to take it a step forward. Just having the plant extract work is good. You will find out that there are 10 plants which are good, but so what? This is not going to go any forward for its activity, except maybe the Ayurvedic. If it's a spice, it could be used, but if it's really a plant that you identified some plant that has really potent activity, it may be very potent, it may be extremely good and not heard of, it should be done. And India has a wealth of information on that, but people have not really capitalized on that. And the way to do that is to generate your own group. So, with the funding, and India has no shortage of funding. United States has no money for research, or there's a lot of money, but there's a lot of competition. Whereas here you can get money. So combine the groups together. Like if you are a cell biologist, combine yourself with somebody who is a medicinal chemist, somebody who is doing biochemistry, somebody who can synthesize compounds, and then you can really make a stride big time into this kind of work. Otherwise, it's a little bit difficult. It's to go forward and find new compounds. So, if you have any questions, I can answer. Because we, while doing this, we evaluated when I left. This was when I was at a University of Illinois Medical Center. We had this program, and I left UIC in 2004. I went back to IIT Research Institute as a vice president and also as a head of cancer division. At that time, they had analyzed 3,800 plants, and out of that, when you write the report, annual report or yearly report to the NCI, and say, oh, we have 2 compounds, right, which are really good. They were thrilled because they said that, you cannot even get 2 compounds out that are novel. You will come up with a compound, you do all these things, and finally you can come up with so I found curcumin. Well, thanks, but that's already known. You have to find something that's very novel. And I'll show you the data that the compound that we found, and we'll go from there. So any questions, I'll entertain. No? So then I think we can call it a day. Start tomorrow at 10 o'clock with carcinogenesis and cancer prevention.